Good morning, everyone. Welcome to today's MPG Primer. We are very excited to have Diane Zhang here with us today. She is a senior in computer science and molecular biology at MIT. Very impressively, she is currently working with the Regev lab at Broad to understand transcriptional variation in tumors, focusing on single cell and single nucleus RNA sequencing data from metastatic breast cancer. Her previous work at Boston Children's Hospital and Harvard Medical School focused on computational analysis of transcription in cardiac disease and liver cancer. We're excited that she will be joining an MD-PhD program at Duke this summer to continue her training. I'm very glad that she is here today to share with us her results looking at this transcriptomic variation in patient samples. So Diane is happy to take questions during your talk. So please type in the Q&A or raise your hand and she'll answer your questions as we go. Thanks so much.
Thank you so much for having me today and for the lovely introduction. So today I'll be talking about Slide-seq, which is the computational method that I developed alongside Dr. Johanna Klughammer in the Regev lab for the past couple of years. Slide-seq is a computational method aimed at detecting spatial copy number alterations from Slide-seq-likespatial transcriptomics data, and I'm excited to share with you what results we have. So the overall problem that's driving our research question is how to characterize the diverse genetic variation that exists in tumors. Over time, as tumors grow and develop, they accumulate mutations, somatic mutations that can either be point mutations or even large-scale copy number alterations that start to confer the cancer resistance against treatment, increasingly aggressive characteristics. These mutations may even be just passenger mutations. But as the cancer divides and as these mutations accumulate, tumors often start to develop subclonal structure, whereby there are distinct populations in the tumor that have genetic profiles with a set of subclonal mutations that exist specifically in that phylogeny or that lineage of that tumor. And so because these subclones are so problematic in the clinic, we were really interested in understanding how we can use genetic tools to understand this diversity in the tumor microenvironment. And importantly, this question is driven around the idea that cancer is a complex dynamic disease where tumors not only exist by themselves, but also exist in a diverse environment with immune populations and normal cells. And so the interactions between different cell types and the dynamic growth of the tumor over time really drive how the genetic landscape is formed and what patient outcomes are like. So recently, there have been spatial transcriptomics technologies that have given us a lot more insight into the environmental context of different types of tissue, including tumors. And spatial transcriptomic technologies kind of fall on different axes. On one axis, there are imaging-based spatial transcriptomics technologies. And then recently, there's been the development of sequencing-based technologies where you can actually profile the entire transcriptome at a near single cellular resolution in a small portion of a 2D tumor tissue slice. And so Visium spatial transcriptomics is one method whereby you can get 55 micrometer resolution. And specifically, what we'll be focusing on is Slide-seq, which offers even more near single cellular resolution, as well as whole transcriptome profiling of tumors. Recently, there has been even more development in the space where we can get spatial resolution at single cell level. But really, we're interested in this question of how can we detect genetic variation from one, RNA-based sequencing data, but also two, spatial transcriptomics data, which offers unique challenges because of this sparsity of this kind of data, even relative to the already sparse single cell data. And secondly, there's the challenge, of course, of inferring genetic variation patterns from RNA-based data. To solve this question of how to extract genetic changes from RNA-based data, there's already been work from the RegEv lab in detecting copy number variations or alterations, which are these large-scale genetic amplifications or deletions from single-cell RNA sequencing data. InferCNV is a method that employs genetic-based or expression-based smoothing of the transcriptome in order to extract wide-level changes in genetic gene expression that is indicative of copy number changes, but smoothing such that you suppress the individual gene level fluctuations that are more small level and that don't really reflect the large copy number changes that you want to detect. However, when employing InferCNV on something like Slide-seq, a spatial transcriptomics technology, the sparsity of Slide-seq makes it really hard to actually extract meaningful information, which led us to develop Slide-CNA. So overall, I'm going to briefly talk about the workflow of how we approached implementing Slide-CNA on various metastatic breast cancer samples. And hopefully at the end, I'll do a short little tutorial about how to start using Slide-CNA yourself on your own data sets. So in our project, we focused on metastatic breast cancer to the liver with 2 individuals. And so these 2 samples can be labeled HTAP895 and HTAP944. Within these 2 metastatic breast cancer biopsies, we were able to get matching Slide-seq data as well as single nucleus RNA sequencing data for reference. And for one of our samples, we also had whole exome sequencing data, which was useful in terms of analyzing a ground truth of copy number profiles with actual genotyping data and not just RNA-based data. For both of these samples, we ran Slide-seq, or we ran Slide-CNA on the Slide-seq samples, as well as InferCNV. And using the single nucleus RNA sequencing data, we were able to run Slide-CNA without the spatial parameters, as well as InferCNV, which is kind of the gold standard for single nucleus or single cell based CNV detection. And then lastly, you can see here that from the single nucleus RNA sequencing data, we generate an in silico spatial data set, which I'll talk about a little bit later. And the purpose of generating this in silico spatial data set was to actually see whether we could detect subclones with our Slide-CNA method, because we didn't really see strong subclonal structure in our own Slide-seq samples. And so we wanted to see whether we could, in silico, recapitulate with Slide-CNA the expected spatial structure of our data. So going into the actual Slide-CNA methodology about how we developed our method, there are 3 major parts of this pipeline. First, Slide-CNA involves calculating a pseudospatial distance in order to bin or group nearby beads or the molecular units of Slide-seq into larger units, such that we're able to get more counts per molecular unit and overcome that sparsity issue. Secondly, is a binning and copy number alteration calling procedure, whereby we divide the binned beads by their the copy number alterations that are called into potential different subclones. And then lastly, again, is clonal determination, where we start to look at the different copy number alteration based subclones and see what kind of differentially expressed genes are associated with them, as well as which kind of gene ontology terms are associated with the clones. But going more into the pseudospatial distance calculation, which is really the bulk of what Slide-CNA does, on the left-hand side, you can see here how we take beads on a 2D dimensional coordinate system and actually calculate the Euclidean distance between the different beads in order to generate a a two-dimensional matrix, or a not two-dimensional, but a bead by bead matrix that tells you the distance between each bead in space. And so that's really important for us in terms of understanding and leveraging the spatial aspect of Slide-seq. Secondly, on this right-hand side of the pseudospatial distance calculation, what we do is for each bead, we take the smooth expression across all of the chromosomes in a very inversine V-like manner using a window of 101 genes to get a smooth expression level, as well as adjusting for normal or reference cells in order to get the malignant cell relative expression, and then calculate the expression of, or calculate the distance of different beads in space by their differences in gene expression. And so you can see here that two beads that are very close in gene expression space have very similar gene expression profiles and most likely come from the same cell type. Whereas a bead that is further from other cells, in this example, bead 3, that's further in gene space is more likely from a different cell type and has a different expression level. And the reason why we incorporated this gene expression based distance metric is because without this, Slide-CNA actually doesn't incorporate the spatial aspect, whereby we expect that close by beads most likely have similar gene expression profiles. And on the latter side, if we only use this gene expression based distance metric in order to group beads together, what we see is that Slide-CNA, or that the clustering is such that beads all over the grid will be clustered together, not really in a spatially coherent manner, which again takes away from the aspect whereby we're trying to extract meaningful spatial patterns while overcoming the sparsity issue. So that's why we need both the spatial and expression aspect in order to bin beads together. And here we weight this, you can input different parameters into Slide-CNA in order to weight the spatial distance metric and expression distance metric according to how you see fit to generate a pseudo distance matrix. And that pseudo distance matrix is what we input into our binning procedure. And so beads are then binned by how close they are together in space and by how similar they are in terms of expression. And you can see here that beads are generally kept close together according to our different spatial and expression weights. And using these bin beads as units, we can then use an inversine V-like procedure to actually call copy number alterations based on elevated kind of smooth expression gene scores to indicate amplifications and the opposite to indicate deletions across all chromosomes. And then once we have those heat maps and those copy number alteration scores, which are based on the smooth expression for the different bins, we can then actually group and cluster the bins hierarchically into subclones based on their copy number alteration scores. And so overlaying that across space, hopefully that'll give us insight into how different spatial units or how CN copy number alterations segregate across space, and also how that overlaps with a different cell type status. And lastly, as I mentioned before, you can then perform downstream analyses on those potential subclones. So was there a question? Oh, sorry, I thought I heard a noise.
Thank you. I do not see any questions, but I will let you know when they appear. Thank you.
So going into our actual data sets of what we have for Slide-CNA, we had 2 metastatic breast cancer Slide-seq samples with matching single nucleus RNA sequencing data. So for our first sample, HCHAP 895, you can see here on the left that this sample does have spatial structure in terms of how non-malignant and malignant cells are partitioned across space, and that hopefully that'll give us more information about, or hopefully with Slide-CNA, we can get more information about how this malignant population is divided across space. On the right-hand side, you can see Slide-CNA's resulting heat map for the sample. And so what we can see is that already in the top blue, Slide-CNA is able to separate non-malignant bins from malignant bins, which is a good sanity check that our non-malignant beads don't have strong copy number profiles, and that even within the malignant population, that there are, in this pink color or red color and green color, 2 different clones detected based on the copy number alteration profile. Down below, you can see the results for InfoCNV on the single nucleus RNA sequencing data, which we use as validation. And what you can see here is that Slide-CNA is able to recapitulate most of the copy number profiles that you see in InfoCNV with single nucleus RNA sequencing data. Meanwhile, if you perform Slide-CNA just upon or if you perform InfoCNV just upon the Slide-seq samples, you don't really get any signal. So this binning procedure actually really improved our ability to detect reliable copy number alterations. The division between the clusters 1 and 2 in the malignant population also have a spatial patterning, which you can see in the bottom left corner across space. So cluster 1 is very much on the outside or on the border of this sample. Cluster 2 is more on the middle section, closer to the non-malignant populations. And cluster 3, again, mostly represents non-malignant cell types. And what this tells us is that although we don't necessarily strongly expect, or although we can't confidently say that there's subclonal structure between 1 and 2, because the copy number alteration profiles are very similar, although there are slight differences. And we also don't know if cluster 2 has cell type mixing from the non-malignant cells in cluster 3, which are spatially close together. We do see that we're able to extract some subclonal variation, or that there's the potential to detect subclonal variation using Slide-CNA. And that, importantly, we are able to detect copy number profiles, even at the extent of subclonal variation. So for example, you can see here in orange on chromosome 6, we're able to see a deletion on this P arm of the chromosome. Next, we wanted to see or investigate the spatial patterns of these copy number changes even more closely. And so what I have here are different bin beads colored by their copy number alteration score, which was generated by that prior heat map. What you can see here on the top is that across a couple different chromosomes, some of which demonstrated copy number alterations, the mean copy number alteration score varies across space. So chromosome 11 and 12 don't seem to have strong copy number profiles, but chromosome 13, 21, and 22 seem to show slight variation in the mean copy number alteration score for that chromosome across space. And particularly in chromosome 22, you can see that the differences in copy number alteration score track well with the malignant and non-malignant spatial cell types. Meanwhile, just below that, what I'm showing here is for chromosome 6, the different quantiles of the copy number score. So 1 indicates the minimum copy number alteration score, and 3 is the median, and 5 is the max, and 2 is the 25th quartile, and 4 is the 75th, the 75th kind of segment. So for chromosome 6, you can see that there is, even within the malignant cell type, a difference in copy number alteration score across space, and that there's a gradient where that such that in the lower left-hand portion, there's a much stronger deletion or lower copy number alteration score signal compared to the right side with the non-malignant cell types. And what this suggests is that Slide-CNA is useful for detecting even gradients of copy number alteration across space. Lastly, we performed differential gene expression analysis on the different subclones or clusters detected from this sample. We didn't really see strong differences in the differentially expressed genes, but we do see that there is the potential to detect differences in gene expression. Next, I'll display the results of HCHAP944, which is our second sample, for which we have slide-seq and single nucleus RNA sequencing data. For HCHAP944, I kind of went over this very briefly in our overall pipeline, but HCHAP944 didn't have strong or reliable non-malignant cell type annotations. And even the non-malignant cell types that we annotated for the sample were widely dispersed among the sample, which made it even harder to detect reliably using Slide-seq, which combines a couple of different cells per bead. So we ended up doing a reference transfer from our previous HCHAP895 sample, which is in this upper right-hand corner, taking the non-malignant cell types from that population and transplanting them spatially offset from our other sample in this upper right-hand corner. What this allowed us to do was have a better set of non-malignant cell types, which is important. And what we found is that this non-malignant cell type transfer didn't introduce any batch effects into our sample, and that we were still able to recover copy number profiles that were similar to the inverse CNV results. And so what you can see here on the right is the heat map generated for this sample, where we have, again, non-malignant cell types partitioned from the malignant cell types based on the copy number alteration scores. But differently from our other sample, Slide-CNA didn't recover much variation within the malignant population. And the division here between the clusters was more within the non-malignant cell type, which didn't give us as meaningful information about subclonal structure, but was still useful in terms of identifying copy number profiles in general for this sample. And as you can see below, for our validation using inverse CNV and the matched single nucleus data, we see very similar copy number profiles. And so this suggests that even with this sample where there was a lot of normal cell type mixing within our malignant population, and where we had to introduce a non-malignant transfer, that we're still able to recover copy number profiles that are very similar to inverse CNV. And again, on the bottom left here, you can see the division of the clusters across space, and that we really do divide out the non-malignant and malignant cell types by copy number alteration. Over here, you can see, again, the mean copy number alteration score at the top for a couple of the selected chromosomes. There is, again, a division across space according to the non-malignant and malignant cell types. And on the bottom here, where we're looking at the quantiles of copy number alteration score for just chromosome 8, and chromosome 8 here is in light pink, where we see an expected amplification in the malignant cell types. You do see that this amplification occurs primarily and much stronger in the malignant cell type population in this lower left-hand corner.
I noticed that there's a hand raised, and I can't tell from my screen immediately whose hand it is. So if you are raising your hand to ask a question, please feel free to unmute or post your question in the Q&A. There is a question in the Q&A. I will read it now.
Thank you.
The audience member says, I saw you cluster CNV bins. Have you ever tried to cluster by CNV segments or breakpoints?
Yeah, so that's a good question. So that's something, let me go back. So how we do clustering here in terms of clones is by, we do cluster by the different bins that we identify, and we simply use copy number variation scores based on the heat map generated. In terms of actually creating CNV breakpoints, that's that would be a little bit more of a complicated analysis. We don't actually call specific copy number variation segments here or copy number alteration segments. Rather, we generate a heat map where you can visually see different copy number patterns. And so in order to implement what you said, we would have to potentially call CNV breakpoints, which is a slightly more challenging problem. And an easier way would be to just look at the P and Q arms of the chromosomes and use those, and instead of copy number alteration scores in order to cluster, but that would also involve calling copy number alterations within a P or a Q arm, which is not something that we've had the chance to look at yet. And then I'll also add that using just pure copy number alteration score can be useful in detecting smaller subchromosomal copy number alterations that we otherwise wouldn't be able to detect. But you are right in that it is a bit noisier than if we just used copy number segments. So continuing on for the second sample, looking at the different clusters and how gene expression varies by them, we can see that clusters 2 and 3, again, tend to have very strong expression levels of these immune-related genes in the middle here, and that these more breast cancer-related genes at the bottom in cluster 1, again, are much more highly expressed in this malignant cluster. And so that's a nice validation that that the clusters are, again, dividing as expected, but really, hopefully, we would see that these differentially expressed genes might suggest something about subclonal variation. So because we weren't able to detect really strong evidence of subclonal variation, and we weren't even confident that there was this kind of subclonal structure in our samples to begin with, we decided to construct an in silico spatial data set to actually mimic subclonal structure and see if our method could recover that. And to do this, we took the single nucleus RNA sequencing data from both of our samples and positioned them in 2 different ways. In one way on the left here is we generated a 3000 by 3000 two-dimensional grid where we separate the non-malignant cells at the very top of the grid to kind of emulate a situation where there's very clear separation between the normal and the malignant cells of your sample. And then we overlay malignant cells on the bottom here. And we titrate the malignant cells or the malignant nuclei such that this nuclei from one sample tend to be placed more strongly on the left side, and the nuclei from the other sample are much more commonly placed on the right-hand side in a gradient manner, such that there is mixing of the 2 samples in the middle. What this does is this allows us to see if, similar to how subclones are formed, these 2 samples we expect to have different copy number profiles. And so what we're hoping to see is whether we can capture these 2 different samples across space because we'd expect them to have different copy number profiles. And then in the middle here, we want to emulate what happens in actual tissues whereby cells mix and overlay across space, and to see whether the copy number alteration scores are strong enough such that we can divide out those 2 different samples, even when they're mixed together in the middle.
Diane, there's another question in the Q&A that I will read now. What is the limit of detection of slide CNV in terms of bin length and coverage?
Yeah, so in terms of bin length, This is actually a parameter that you can modify in your own sample or in your own iteration of Slide-CNA. But what we found best was that Slide-CNA works best on about 12 beads per bin. And again, this is such that you want to have enough molecular counts in your bin unit, but you also don't want to have very few bins because, again, you lose some of that spatial structure, and there's this constant offset between sparsity and resolution in terms of space. And so if you bin more beads together, you get more counts per unit, which gives you more computational power, but at the same time, you lose a lot of that spatial resolution. And a lot of the benefits of using Slide-seq over other methods such as Visium are that you get this near single cellular resolution and can start to infer or characterize even more intricate aspects of whatever tissue sample you're using. And in terms of coverage, as you mentioned, we found that we actually investigated this a little bit. I'm not going to share the results here in our in silico spatial analysis, but we see that Slide-CNA has about between 500 to 1,000 counts per bead, and that is generally enough such that Slide-CNA can generate meaningful patterns. And if you start to go below that, you lose, you just don't have enough power at all, and the data are too sparse to really get any signal even with Slide-CNA. In terms of if you're implementing Slide-CNA by yourself, I might suggest that if you have a little bit more higher coverage for your sample, that you can reduce the number of bins that you input for each bead because you have more power to your data already. And the opposite would be true if you had even more sparse data. You might want to increase the number of beads per bin that you use. So for this in silico spatial analysis, again, we have on the one hand, this simulation where we're separating the non-malignant nuclei from the malignant nuclei, which we have in this overlaying gradient manner. And in the second hand portion, we directly overlay the non-malignant nuclei right on top of the malignant samples to see or to test the effects of Slide-CNA in this situation that occurs very often in tissue samples where you have a lot of mixing between your reference and your cell type of interest. From those two single nucleus RNA sequencing data kind of spatial grids, we then generate in silico beads that would kind of emulate how Slide-seq captures a certain number of, usually like anywhere from 1 to 10 cells per bead using a Gaussian kernel. And so using this, we're able to get a new matrix or a new two-dimensional grid with different beads and their maximum cell type label, whether it be non-malignant, HTAP895 or HTAP944, as well as their counts matrix. Here you can see an overview of what that pipeline looks like for the two different situations that I mentioned before. For the non-malignant nuclei separation, you can see here that the bead generation works pretty well and that we're able to maintain the non-malignant nuclei separation, as well as the two samples maintain their gradient-like overlay across space, and that the total number of cap reads is mostly consistent across space. For the non-malignant nuclei mixing, you can see a very similar situation where we just confirmed that our bead generation, our in silico bead generation is effective. So from this, showing the results for what Slide-CNA looks like on the non-malignant separation case, you can see here that we are able to get strong copy number profiles that actually match the expected profiles from the infer CNV case. And actually, to mention before, importantly, it's to note here that we actually downsampled our nuclei to 10%. And downsampling our nuclei to 10% gave us number of reads per bead around a little bit under 1,000, which is about the same level of Slide-seq. And so we're able to see that even when we downsample our single nucleus RNA sequencing data to Slide-seq levels in this in silico simulation, that we are able to recover, number one, good copy number alteration profiles. And number two, we're able to actually identify this subclonal structure that we had imposed prior to this analysis. In the light green here, you can see that HCHAP944 is mostly associated with cluster 3 that is called based on the copy number alterations of the sample, and that these profiles are mostly consistent with the inverse CNV results. And on the bottom here, even though there's a slight, there are some binned beads that are from this HCHAP944 sample originally, that mostly this purple cluster or this pink cluster associates with the purple HCHAP895 beads. And spatially, you can see this on the upper right-hand side. The non-malignant beads partition out at the very top, and in terms of the malignant beads, the two quote-unquote subclones are called or are separated based on Slide-CNA, with HCHAP895 on the left and HCHAP944 on the right. And even in this middle section where there was this mixing between the two samples, we're able to see that this doesn't confuse the analysis as much, and we're able to still separate the two samples for the most part. Meanwhile, for the non-malignant separation case, what we see here is that even when we downsample to 10%, that we don't really see a strong malignant or non-malignant cluster separated out of our clusters, which actually is expected because when we bin beads, it's very hard to, or when we bin beads, and even when we generate the in silico beads, we're mixing the non-malignant and malignant cell types. So it's a bit harder to cluster out the non-malignant population, but we're still able to see strong separation between our two samples, HCHAP895 and HCHAP944, based on copy number profiles. You can see here that there were 3 clusters called. One cluster is in pink, which mostly associates with this HCHAP895 sample in the left-hand side, which you can see on these spatial axes over here to the right. And then interestingly, what happens is that there's a kind of middle portion here in blue, which is composed of both HCHAP895 and 944. That represents this middle portion where there's cell type mixing. And so what this tells us is that it's very interesting to see that when you combine copy number profiles in the middle here, that you don't really get you generate a copy number profile that is kind of unique from the prior two. And what that looks like is very noisy data at the bottom here. And so this suggests that Slide-CNA maybe doesn't work so well on very mixed subclones if there's not a strong non-malignant reference present. However, we do see that on the very right-hand side, when there are mostly HCHAP944 pure beads and pure bins, that we do see copy number alteration, a separate copy number alteration profile that is similar to the inverse CNV results. And again, the number of reads per bin is very similar to Slide-seq data. And so overall, what this in silico spatial analysis tells us is that Slide-CNA has the potential to recover subclonal structure from Slide-seq-like data, and that potentially the level of mixing of non-malignant nuclei or non-malignant cells matters a lot. But that Slide-CNA is still able to operate in both situations. So now I'll go into a brief tutorial about how to actually implement Slide-CNA by yourself. So the code for Slide-CNA is available on GitHub, and the Slide-CNA vignettes will be available on GitHub at this link very soon. Not quite yet, but I'll go over a brief tutorial in the meantime. I'm going to stop sharing my screen and instead share my Jupyter notebook. So here I'm demonstrating a workflow for Slide-CNA using HTAP 895. So what Slide-CNA does is it takes, it uses one major function, which is, I'll show at the bottom here first to give you an overview. It uses this run Slide-CNA function, which involves a serial object that takes your Slide-CNA data with its counts, as well as this metadata over here. And this metadata is important because it contains all the different beads, their X and Y coordinates across space, and also cell type annotations. So we require that you annotate your cell type or your different beads for malignant or non-malignant prior to this analysis. And in the paper, what we do is we use Seurat clusters in order to manually see, based on a module of metastatic breast cancer genes, to see which Seurat clusters tend to be malignant and which Seurat clusters tend to be non-malignant. But there are different ways of annotating your sample. And what actually I've demonstrated here in this tutorial is using RCTD if you have a single cell reference to generate cell type annotations, as well as cluster type, which is malignant or non-malignant reference or annotations. And so here we take in a filtered and processed single nucleus RNA sequencing data set, as well as a processed Slide-seq RNA data set. What we do from that is generate RCTD annotations from this, and we get normalized weights for each of the beads according to their different cell types. So what robust cell type decomposition does is, using a reference, it's able to extract the different, or infer, it's able to infer the different cell types that compose your bead, which again, in actuality, is comprised of multiple cells and multiple cell types, and weight the different probabilities of what that bead is most likely composed of, or what cell types that bead is most likely composed of. And we take the maximum probability cell type for each bead and use that as an annotation of the cell type. Using this, we're able to construct a data frame, which is the metadata of the beads, their spatial coordinates, and their different cell type labels according to RCTD. Separately, we generate a counts matrix, which you should have from your own analysis. And from this raw counts matrix, we're able to filter, or we filter the counts matrix such that we only take cells with over 300 counts across all genes. Using this, we just quickly look at some, marker genes. For example, here CD represents some immune markers, and we can see that they correspond roughly to the non-malignant cell population across space. From this, we generate a Seurat object from our counts matrix and our metadata with the beads, and then we label based on the cell types generated by RCTD, normal and malignant. You can see here that the normal and malignant cell type cluster types for this sample, again, are very separated according in Tisney space, but that also, when you look at the actual spatial coordinates, that there is also this separation between the cell types. And using this, we also look at different markers of the different cell types. And this is just an alternate way to annotate your cells for malignant and non-malignant status. And then lastly, over here, you can see that we take the resulting serial object that we generated, the metadata that we use based on RCTV, as well as a list of genes and their location on the different chromosomes, which I'll also have available in this vignette coming soon. And we take those three inputs and put them into run Slide-seq-DNA. And here, this last parameter where you see spatial equals true indicates whether you're using spatial transcriptomics data or not. Slide-seq-DNA also works on single nucleus or single cell based data. It's just that in the pseudospatial distance generation for bead binning, you just won't include any of the spatial information, and you'll only cluster beads or cells based on expression. And there are different parameters to run Slide-seq-DNA, including the number of beads per bin that you want to input. You can change the different weights of how much you want to use the spatial aspect in terms of binning versus the expression aspect in terms of binning. I have different defaults based on what has been best for current Slide-seq data, but if you're running this on other types of Slide-seq like transcriptomics, spatial transcriptomics data, you can modify the parameters. And with that, I will finish this talk, and I'm happy to take any questions if there are any in the meantime.
Thank you so much, Diane. That was a really amazing talk about a really useful and productive tool. I have a biology question, and before I ask that, I just want to encourage anyone else in the audience who has a question to please post it in the Q&A, and I will voice it before we end for the day. I think one of the many really interesting things about this approach is the opportunity to look for broader, I guess, spatial patterns across tumors and across individuals. And I wonder if you've found that there are broad things about like the distributions of malignant and non-malignant cells within tumors that are congruent across individuals, or whether those ratios are broadly consistent across tumors, or whether there's no pattern at all like that if you apply this to many individuals or tumors.
Yes, so far, we've only applied it to a limited set of samples. So I've shown 2 samples here. We've run this for about 4 samples in total. I think in terms of speaking about what this represents about tumor biology in general, I think it comes to show that, one, our analysis of or our understanding of tumors can be quite limited by the technology, or often is constrained to the technology from which we view that. So even Slide-seq involves pretty small, like tissue slices, about 3 millimeters. And so we're only capturing a very small portion of the tumor, but you can already see that there is heterogeneity between the malignant cells and the non-malignant cells. Sometimes these 2 populations occur very closely together. Sometimes they're separate across space. You can see that in one of the samples, there seem to be like slight gradients of copy number alterations across space. But in the other sample, we didn't really see strong spatial patterns in the copy number alterations. I think that hopefully this analysis can be performed on other samples to see other types of interesting biological patterns. But yeah, this is kind of suggestive that tumors are quite diverse, and there isn't really a rule to how they are laid out organizationally, but we hope to characterize some of the different ways that these cell types occur across space, using Slide-seq-DNA from a genetics point of view.
Thank you. And just one more reminder to anyone in the audience who has a closing question to please post it now. Otherwise, I don't see any questions right now, so I guess we will close there. Diane, thank you again so much. That was really, really exciting.
Diane, there was one raised hand. I'm so sorry.
I'm so sorry to miss that. Thank you for noticing that, Sarah. If you have your hands raised, please unmute yourself and we will look forward to your question.
Thank you. Thanks. So very nice talk. I'm pretty interested in this spatial copy number analysis. Yeah, so as you may know, we can also infer the phylogeny based on the RNA expression data. So or whole exome sequencing data. I see you already have that kind of data. Have you compared your subgroup data, subgroup profile versus or clustering data with the profile or pattern get from the RNA get from the expression or for exome sequencing? Yeah, and that's one. Another question is about the denoising. Yeah, I saw there's still some noise or some high level number high level number events in the non-malignant samples. Have you ever, or do you have any plan to denoise them or use other algorithms, something like that? Yeah, that's all.
Yeah,
Thank
for
you.
sure. Good question. I didn't share on the whole exome sequencing, but I can pull them up quickly here. In terms of whole exome sequencing, we were limited to one sample. But let me share my screen. For the whole exome sequencing data, this is very kind of an output from Absolute, which is the copy number detection tool that we use for this. Here, it's a little bit hard to read and compare against our own data, but on a more side-by-side comparison, we can see here that with this whole exome sequencing data, which we use as our ground truth, that when we manually annotated for copy number profiles, we see that at the top here, when we run Slide-DNA on Slide-seq, that we get mostly the same copy number profiles as we do with whole exome sequencing. There are slight variation. Some of that is due to the fact that when running these methods on different samples, you do expect there to be maybe imperfect copy number scores if you're using completely different samples just because of subclonal alterations, but also because Slide-seq isn't perfect or Slide-DNA isn't perfect and uses expression-based method in order to extract genetic changes. For example, you can see a difference here on chromosome 21 between Slide-DNA and Absolute. Also on chromosome 16 and 18, it's not calling these two deletions that we'd otherwise see with whole exome sequencing data. And then to answer your second question, which was about the noise level in the different samples, and I'll just pull up my PowerPoint again. What I think tends to be true for our samples is that when you have less reliable cell type annotation from malignant and non-malignant, you tend to get a bit more noisy results, especially in this non-malignant population up here. When we ran this with RCTD, we saw a bit cleaner copy number profile. So I think part of it has to do with the fact that when you have non-malignant cells that aren't annotated as well, you pick up some malignant cells, and so that can impact the copy number score, because the copy number score is relative to your non-malignant gene expression scores. And then secondly, also, this is true when you bin the different beads together. If you have a lot of, or actually, I think it's in the sample, yeah. If you have binning between non-malignant and malignant beads, you're also mixing cell type profiles, and so that can also add to the noise when you're taking true copy number signal and mixing that with non-malignant or no copy number signal, you can get noise. And so the results are a little bit cleaner in this analysis, where we had single nucleus RNA sequencing data with better non-malignant annotations, but you're right, it's still a little bit noisy. And we haven't worked on necessarily the process to denoise that, which I think also relates to the question before about actually calling copy number alterations and their breakpoints. And before we finish, I also forgot to acknowledge the many people who contributed to this project. And I want to highlight Nikhil Wegal, Aviv Regev, and then Johanna Klukhammer for being mentors on this project.
Thanks so much.
Thank you,
Diane, for an amazing talk. I think we will end there in the interest of time, but thank you again, and we wish you well as you transition to the next stage of your career. And we look forward to hearing more about your ongoing work.
Yeah, thank you so much for having me.
Thank you. Bye.